Okay, so first, my name's Todd Valerius. I am at the Brigham and Women's Hospital and Harvard Medical School. The consortiums I represent, first, I'm PhD trained as a developmental biologist, and I'm involved in the GoodMap project, the General Urinary Molecular Anatomy project, and the Rebuilding a Kidney project. I also serve as external advisor to the Kidney Precision Medicine project. So these consortiums I'm actively involved in. And I'd like to hear from the rest of you. We can go down the list, I guess, alphabetically in terms of the videos. So Kristen, you would be next.
Hi, sorry, this is Kristen. I'm from the Broad. I led with a couple of people on this call, the metadata workshop yesterday. As I said, I'm at the Broad Institute. I've been working extensively on the GTEx project and now leveraging that into Human Cell Atlas projects.
Thank you. And then Laura.
Hi, I'm Laura Clark. I work at the European Bioinformatics Institute, primarily on the Human Cell Atlas data coordination platform. And we're starting, well, the HCA is different from other projects in that it isn't sort of single funder, consistent tissue collection, people wanting to discover samples that are being collected and things. So I figured this was a useful place to be.
Thank you. And Marianne?
I am Marianne Martone from UCSD. I'm part of the BRAIN Consortium, and there's not a lot of sample sharing at this point, but we have been working on, in another project, unique identifiers for sharing biosamples. That's why I joined.
Thank you. And Melissa?
Hi, yes, I'm Melissa Cook. I'm a contractor with the National Cancer Institute, their Information Technology Division, and I specifically sit with their semantic infrastructure team. And I work with the Human Tumor Atlas Network, and I see my two co-moderators from yesterday's clinical data session on here, so I think some of the topics are related. I also work with the Cancer Research Data Commons and the team that's coming up with the harmonized model for that project, and so the biospecimen information is a key part of the puzzle for both HCAN and the CRDC. So.
Thank you. And Sarah.
Sure, I'm Sarah Mazzilli. I'm at Boston University, so I'm also part of the Human Tumor Atlas Network. My group particularly is focused on lung biology, and so lung pre-invasive disease. And so also was part of the clinical metadata moderation. So hopefully that'll be part of the topic today, and thinking about how maybe we could develop into a soapbox idea around this.
Great, thank you. I think I have everybody on, I've covered everybody on video. Anybody else need to chime in who's going to be an active participant? I think I got everybody. Okay, great. First, I wanted to say very quickly, because this is a topic that I do care about a bit, is that I'm very happy that I am in the minority as a male on this call. So thank you all very much for being here. You know, throughout my career, I've always known very brilliant women in science, which is what has attracted me to this field in general, is that there's for me, it feels, at least, there's an attempt at equality. So I really appreciate that. I'm going to share my screen and open up the doc so we can get started. So I share the right one. I'll try to keep the interruptions from my 5-year-old to a minimum as we all adapt. Okay. I think
if you want to recruit scribes from the group.
Thank you. That's the next thing.
Sorry.
If we can have a couple people volunteer to contribute to scribing and capturing our talk, our conversation, that would be great. Anyone?
I'll help.
Thank you.
Yeah, I'm also happy to help as well.
Great, thanks. And everybody's welcome to, of course, open the doc and add things and edit as we go along, which worked pretty well in some of the meetings I was in yesterday. So for this section, what as the moderators originally started to set out, we thought a lot about sample sharing, not only data sharing. A lot of discussions about metadata, et cetera, from yesterday included conversations about what is the right metadata, what do people need, et cetera. But another part of all this is also, I feel, is sample sharing. This is something that's come up quite a bit in the Kidney Precision Medicine Project, where you have a consortium that has 6 recruitment sites, and you have a different set of 6 to 8 analysis sites. So biospecimens themselves need to be collected and moved around. So a lot of work went into how to do that. Some funding was dedicated to actually working out that system. And I think that that's an area where a lot of consortiums haven't needed to spend much time thinking about as they've gotten going. It's all within consortium. So I thought it might be good to sort of touch on that a bit on the sample side, as the data side sort of is the dominant part of this whole meeting the last 2 days. But has anyone here had much experience on the sample side, biospecimens or managing biospecimens, for example? I'd like to start with that, if that's okay with people.
So I can talk more from the experience of collecting metadata and facilitating metadata transfer alongside the samples that are shared. In that, obviously, I think, as we all recognise, that's very important, because that metadata must be carried alongside the data that is generated from those samples and ultimately the analysis of the data. But I know within the UK HCA initiative, there are 2 organisations that are biobanks. Unfortunately, I don't see either of them represented here today, but one is the Human Development Biology Resource, and that's a very long-standing resource that gets fetal tissue. And at least within the UK and elsewhere, I think they do ship internationally for places that are that can have the ethics to have to do experiments on fetal tissue. They send fetal tissue to people, so anybody who's doing developmental studies who doesn't have access to fetal tissue from their own sources can get it from there. And then the other is an organisation that's being set up at Addenbrooke's in Cambridge called the Cambridge Biorepository for Translational Medicine. And they're essentially working alongside the Cambridge transplant teams and get a small window of opportunity when a deceased individual's having organs collected for transplant to also collect material for research purposes. And I think they're starting to, they're shifting. It was previously just research collaborations that allowed them to share data, but they're trying to shift to a sort of tissue bank style footing, and then people approach them with their own ethics, not having to sort of involve them in the collaboration. But I think for consortia like the Human Cell Atlas, which is much more bottom up, if you've got some funding from somewhere and you want to be involved, you can be, that style of tissue bank becomes quite important. Because whereas I think some of the NIH initiatives, people are funded to do the sample collection and to do the data generation. And so I don't know in the US how much there are of these sort of tissue banks that can as standard distribute tissue, as opposed to someone being part of a research collaboration and then they can collect tissue, whether it's waste material or from surgery or biopsies, or sort of specifically collected.
So this is Kristen. I'll just follow in from Laura. And I think it's, regardless of the model, I think it's absolutely key that we discuss this and enable it. We had a wonderful example. Well, actually, I've been involved in setting up the biobanking facilities at the Broad, which I no longer oversee. But just in the 2 projects I've been involved in most recently, in GTEx, everything was standardized, kind of the way that Laura described. And to avoid experimental batch effects, all the samples came to one place. We did careful sort of batch mixed isolations of those, and we kept the same protocol for sequencing. Now, we followed that and got engaged in a TopMed project that was running through the Broad Institute and through another sequencing center. simultaneously. And we were shipped samples where inadvertently they thought it was best to ship samples from 2 different longitudinal time points separately. So even within a center, those went on 2 different experimental batches and had massive batch effects. And then we wanted to be able to compare data between the 2 centers. Now, ostensibly, both centers were running the exact same sequencing protocol and in theory, the same downstream pipelines. In fact, we went to a point of spending about a month completely harmonizing the analytical and QC pipelines. And there was still a massive batch effect between the 2 centers running effectively the same protocol and identical downstream analysis. So I think it's really key, and those are just 2 RNA sequencing protocols, but in where we are right now, where protocols are being developed and modified and changed all the time, I think when you feed it up into a DCC like Laura's or a big data set, to understand these batch effects is crucial. And the only way to do that across some of these technologies is to basically share the same baseline sample. So I think working out ways to enable and encourage this is very important. The one project in the US I know that has it built into its mandate is the HubMap project, where investigators are required to bank additional specimens to do tech dev across new technologies and across others who are collecting different samples. But I think it needs to be more widespread than that, because I think it's really key for us to be able to harmonize and integrate these data and to see what the batch effects are at the experimental levels.
I know the Human Cell Atlas has the standards or samples and technologies working group led by Holger Hearn and a couple of other people who are doing a couple of sort of benchmarking studies. One initially was around, I think, the RNA-seq in different places, and they had a mixture of different cell types. In fact, it's 3 different species, so they could have really good truth about what should be there. And they've done one more recently on cryopreservation, again in multiple centers. I'll see if I can find the preprints and I can add them to the document. But it is something that the Human Cell Atlas is trying to do as well. And I think they're sort of finding solutions for it. It's a shame I don't think we have anybody from that side of things here with us.
I'm actually part of that working group, Laura.
Oh, well, there you go. Fair enough.
So you're right, but some of the experiments to this point have been benchmarking the technologies, and in the context of doing that benchmarking, we've been sharing samples, but that doesn't sort of get this push out to what Todd was talking about. I think this broader sharing of samples, which I think is a really key mandate.
Yeah, I'd like to add that the Kidney Precision Medicine Project also, as part of its mandate, is required to share samples, and they go to a centralized biobank. And these are research kidney biopsies. So they're extremely rare and precious. So the testing on how to move samples around and coordinate and what steps you do what for a biopsy, they used transplant biopsies. Nephrectomy sample biopsies. So those are readily available. It's not healthy tissue per se, but there's an example of how you can use sort of a proxy tissue that approximates something to work out a lot of things before you get to the very valuable samples. And I think that not only is it an agreement on how to do things. For example, they found that if you isolated nuclei from frozen sections or from a sample, the second time that biopsy was moved and then subject to recutting, they had lower yields. So those little nuances that we all know from being in a lab and doing this work don't always make it out to the fore. And I worry about documenting best procedures for all of this. When you talk about metadata and working groups, are people generally starting from a common place and then evolving to their needs, a metadata or SOP approach in each case? Or is everybody sort of starting a grassroots movement to develop what they need?
I could address two things. One is I can come back to talking from the HTAN perspective on the biobanking, but a little insight that I have from both our session on clinical metadata as well as another session I sat in. I think people want to start with something central, but don't always. Some are relying on some of the more new, different consortia, like for HTAN, for example, we did look to the GDC as well as looking to the HCA for places to look for metadata. The hope was not to reinvent or redefine metadata. So I think that that's there, but in some of the smaller consortia, it seems that maybe that hasn't quite gotten to be the case. But I think based on conversations and looking through the outputs of yesterday's meeting, that's a real want, but isn't quite there, and how to mobilize that and make that something that is an agreed upon standard for certain things, I think isn't in existence just yet.
So that,
oh, go ahead.
Oh, sorry, I was just going to pick up on that theme. I sat in on the clinical or the metadata schemas and ontology session in the afternoon, and we had a really good discussion at one point around this topic of how do you get people to come to agreement and to use common standards. And I think it's, you know, there's the knowing that they're available, and how do you get that information out there to people? Part of it is an education and training issue and promotion of what's available. That's one piece of the puzzle. I mean, the other is to get people on board and to have some sort of carrot to get people to adopt common metadata, whether it's semantics for how you name and organize the information, or whether it's about metadata about a process or protocol. So, you know, in some places they've had to use a stick, but this community would probably be served better by using a carrot, right, to get people to adopt common standards. So this theme, I just wanted to say, is coming up in other sessions as well, is how do you get people to come to agreement and share common metadata so that you can do your science and share your samples in this particular case.
Melissa, that's a really good point. And I think one of the valuable things, actually, if we can work with the large and central tissue providers to make sure they have consistent representation of metadata and get their metadata into a database like, you know, either the EBI, the NCBI biosamples, or if there was something, you know, oddly enough at the EBI, I would advocate for one of the sample archives, but it doesn't have to be that solution. There are other ways of doing it in a place where there is a consistent name, where there is a set of standards, and then the scientists who are getting data from those repositories, all they have to do is go with the name to the database and they get all the metadata they want. They don't have the work of transferring that metadata. It doesn't come on in a PDF or a printed manifest associated with the, you know, the cool box that the sample was delivered in and has to be transferred into their own LIMS. There are lots of upsides. Of course, it's how you get enough people to put in enough work that then the upsides are realized, because the early part of that process is not straightforward, and it does take effort to get because sometimes these tissue banks are small entities themselves. They don't have a lot of resources, particularly resources of people who are computationally have computational expertise, as opposed to, you know, the important anatomy or sort of tissue processing expertise. But I think sort of you mentioned protocols as well, and I think Todd, you also mentioned this. I don't know how much people are adopting protocols.io, because protocols.io is the sort of place where, in theory, at least the HCA is meant to be sharing its protocols. It has both public and private sharing, and it almost has a sort of GitHub sort of software style. Someone can fork a protocol and so start with what someone else suggested. And when I first heard about this, it seemed like a great way to give everybody standard SOPs for sample collection and sample processing. But I think at the same time, everybody's trying to figure out how to do it independently. And so we're still ending up with a bunch of independent protocols, and nobody has the capacity to try and resolve them and bring them back together and do the sort of standardization that Kirsten was talking about, you know, and whether, so it's who would we need to drive that sort of effort, and is that sort of effort worth it? Or, you know, if you talk to the analysts, as long as we capture how it was done and whether it was which protocol it was done by, does sort of statistics solve it in the long game, you know, sort of in the end game? Because there are so many different covariates that trying to, is it worth the effort to overly standardize that compared to the value you get? And again, so this is where I haven't worked in a lab for a very long time, so my knowledge of how influential the lab techniques are is pretty poor.
First, first I just want to apologize for dropping off as my desktop rebooted, which is always pleasant. That's a good question. I want to ask about protocols.io, because the fact that there are limitations because it is a for-hire service. So is that the best place for, it's one of the topics we were hoping to cover too, is that the right way to do it, or is it the best way to do it for labs that don't have the ability necessarily to expand? Once you get to a certain point, you need to pay for access.
So I will be honest, I get your discomfort with it being a for-hire service. My understanding is, at least for HCA members, is because CZI is part of their, I suppose, funding model, that in the cell atlasing world, there's a lot of interest in collecting this data and giving people ability to use it for free. Because the reality is, if you sit down and talk to Lenny, his profit mode isn't getting academic labs to pay to use the service. It's getting the reagents companies and the technology companies to pay him for insight into who's using which reagents and who's using which technologies. So the academics are not Lenny's source of profit. So I think negotiation is always possible. And of course, again, depending on how private you want your protocols to be, I think if you're using the public space, it is free almost. I think the whole stack is free. It's if you want private sharing that there may be a charge. But I think, at least for the HCA, if you're happy to share within the HCA, and I don't know, I have never been involved in the conversations about contributions as to whether equivalent organizations could get equivalent agreements and who would be paying for that. What would be the sort of dollar figure on those agreements, or whether it would be done, because reality, that's not his profit model.
No. And I think we do approach the for-profits with a little bit of mistrust, you know, given the history of scientific publishing. But the fact of the matter is, these resources do have to be sustained. And a lot of them, and disclosure, we have one of these ourselves, where we're trying to, you know, build sustainability models for these types of entities. Usually, as Laura says, you start this so that you can work with commercial companies, because it's a lot easier working with commercial companies when you are a commercial company than if you are a not-for-profit or an academic institution. And as long as the governance of these organizations are transparent, I think we do have to have some sympathy for the fact that these things cost money. And he is trying, as Laura says, as best as possible to keep this free for academic use. It's not a big, they're not going to pay a lot of money for it anyway. It's hard enough to get them to put their protocols in there. So I think it's possible for these sort of consortiums probably to work out deals. I doubt that it's exclusive to HCA.
Yeah, I think they have a very nice system, and I do. It's not that I'm uncomfortable personally with working with a company in any way, but it does go to sustainability. I think what I'm leaning toward is, this is an area that should be funded by NIH, for example, or other government agencies, and it's not. There's an expectation that things are shared, but a nice system that's actually robust, like protocols.io, or even what Nature has been doing as well for a long time. Those do cost money, and there does need to be money dedicated to that.
I can add for HTAN, as we are currently prescribed to use protocols.io, both for kind of collection as well as any processing related. So to my knowledge, there's no cost associated with it at this point, and it may be due to, again, negotiations that are outside of my scope with Protocols.io to utilize them for the consortia. Just to really quickly jump back before we move forward, as far as the biobanking efforts in the HTAN, just to kind of bring everyone up to speed there, I think there's two pathways for this. So mostly they're funded through these trans network projects that are they're set aside fundings from the NIH that are meant to encourage collaboration across the network. I don't think there's been a tremendous amount of communication for outside of network sharing, but I think certainly something I think that will come up as the consortia continues to grow. And so there's two ways in which we are getting samples. There's the CHTN, the Cooperative Human Tissue Network, which is outside of the HTAN. It's a pre-existing network that the NCI supports for kind of collection of both diseased and non-diseased tissue in the United States. And I'm happy to, I'll throw a link in there in just a minute so people can check it out. So I know we've gotten some samples from there. Peter Serger's group at HMS is leading some efforts around this, where they're using this tissue along with others to standardize imaging or protein-based staining protocols across a number of institutions. And so similar to, I think, some of the other mentioned kind of standardization using a standard piece of tissue to test different methodologies, whether antibodies or otherwise. So that's one way. Another way, I saw my colleague Asaf was on here earlier, is there's a homegrown effort to do this with a focus on kind of smaller tissues. As I mentioned before, our group works in the pre-invasive space, and so we're working with pre-invasive biopsies, also very unique, hard to get and limited in the tissue quantities, but we're working to try to see if we can bank up some of those as well as other tissues from across other sites. And so this is, again, a different trans network approach, but to try to build a biobank. And right now, we have some ideas of, again, based on validating methodologies used at different centers, but I think there's a want to have other uses for this tissue to provide kind of what happens if we don't use up all the tissues or we don't have a project associated with just yet. But I think there's, again, interest, but right now, inter-network, but certainly potentially opportunity for working outside of that.
So is on the grounds that, if I remember from my own moderator briefing, because I was moderating a session yesterday, we're meant to be defining problems. Is a good problem to write down here facilitating cross-consortium discovery of samples? So where a consortium has collected potentially a rare sample, and for whatever reason they aren't going to use all of it, and they want to flag it for access, that they can somehow tag. They can somehow tag samples that is still available. Is that? I mean, would that be a good problem to discuss? Helping, and so it's actually discovery of samples across all of these cell atlasing initiatives that are available. I mean, I think the other angle of this, and so we're sort of working in two ways. One about sample discovery with the Addenbrooke's resource CBTM, because they freeze samples when they aren't giving them. If they're allowed to take them from a donor, they don't want to see them wasted, obviously. But HDBR, obviously their samples are very small, and it tends to be when they get a sample, because they don't get more than 1 or 2 a month, that it's all used, but they want people to be able to do a sort of, okay, these are the sorts of samples they get. It's reasonable for me to write an application for a sample like this, because they could provide it on, whenever, wherever I am in the queue for priorities for when these samples happen. Because obviously, yeah, in the instance of these are essentially aborted fetal samples, they're not, well, I wonder, actually, I suspect they're probably not substantially less common than deceased transplant donors, but yeah. And that's sort of two different use cases on sample discovery. Can a resource get a sample that I'm interested in? And actually, is there a sample, a frozen sample, or a fresh sample that I could use right now for the experiment that I'm planning?
I think that's a great idea, and I think the first part of our conversation goes to what types of information need to be captured along with that, so that there's a standardization of the metadata associated with a biosample, and then do people want to participate, at least for a portion of their collection, in a centralized discovery way. And what would that mean underneath? Would that mean that there's a funded way to develop a clearinghouse or portal for discovery, or one of the existing ones could duplicate their software stack or their approach and be funded to actually serve as the data sharer? Are there instances where this has worked elsewhere?
So the biosamples database at the EBI has acted as the sort of central store of metadata associated with a few cell line projects and a few projects for, I don't think necessarily for human biosamples, but for plants and animals. And essentially there is a sort of tag system, a project label that is used in that environment, and then it's very easy to pull up a version of their search interface that just targets those samples. And so that almost comes for that. All we need is a metadata standard that we could ask them to enforce, and there are building solutions to do that, because we're doing that for the Human Cell Atlas. So there is at least from a minimal viable product perspective, there are existing solutions, and they do mirror samples with the NCBI biosample database. And so if a particular organization had a mandate to use the NCBI, the challenge of course will be if people want to keep sample records private for a certain period of time. And I don't know what I suspect private samples aren't mirrored, and whether you'd want, obviously my personal perspective, it's better if these things are open, but I appreciate sometimes people are sensitive about exposing details of their own plans by exposing the samples that they're using.
Well, I would say not only that, but also you want to finish your work, okay, and then you might want a set of samples that you want to essentially donate access to, maybe put it that way. And then you would want some reassurance if it's patient data on how that's going to be handled. Every country has a little bit different approach to patient privacy. So I think the first response from somebody who has a biobank would be concern about that.
So to address, I mean, the when you publicly release information about your biomaterials, I think there are ways of addressing that potentially. If you have common standards and people adhere to those standards internally, and then when things get exported, it all works. From a what data can be exposed from a sort of ethics consent standpoint, the biosample database at the EBI is already working with the Global Alliance for Genomics and Health and the EGA to use metadata standards. So the information that can be publicly released and publicly indexed is, because we won't facilitate cross-consortium sample discovery unless there is public metadata, because nobody's going to go through a 6-month application process just to see if a sample exists. But the stuff that is private, the clinical information or the phenotype data that cannot be publicly released, could potentially be stored within a database like the European Genome Phenome Archive with a data access committee in an appropriate standard. I think they're phenopackets is what they're trying at the moment. So that information was still accessible and was still accessible programmatically, but only to the people who are allowed to access it. So there are definitely sort of nascent solutions to the public, the sort of privacy from an ethics standpoint perspective. And I think there are solutions to the privacy from a competition standpoint perspective, but they are just slightly more, slightly less seamless.
Have others, go ahead, Kristen.
So I like this idea of trying to pull together what's there in a search, although the more we talk about it, the more we come upon the complexities of sharing. We haven't even touched on MTAs, things like that, shipping to different countries and so on. But what about something simple, Laura, which is in terms of ingesting data, say for the Human Cell Atlas into the DCC, what if one of the required questions is simply an answer to for this data I'm uploading, do I have any residual biospecimens left that I would be willing to share and people can reach out to me? What about even starting with something simple like that so that we might know if there's residuals left?
Yeah, I mean, I think that would be a reasonable thing to have. And I mean, I think you've got two angles on that. From a within consortium, just a project is labeled, this is likely to have residuals. The challenge will be is how do you make sure they let us know when the residuals are all gone? Because after a certain period of time, they will either all be gone or they will have been in storage for too long or something. And it will get frustrating for people to ask. But certainly that could be one of the other things that came up in our conversations yesterday was Sarah Teichmann was part of the working group with him. She was saying for the excellent COVID-19 work that they're doing, it's been quite challenging to find who has pre-publication data so they can approach to even just get the expression levels for those 3 genes that they're super interested in. And again, if we had some kind of project registry, and a project registry that could have this project might have samples to share or is willing to engage in sample sharing. I mean, the sort of broader model and the model that we've used for IPSC projects and for other initiatives has generally been where there's been a tissue bank. And obviously tissue banks are generally set up to establish MTAs quickly and to have sort of standard paperwork to enable that to go. Whereas if it's a hospital doing a collection as part of a research agreement, you're right, giving those biomaterials to a third party potentially takes a long time in terms of lawyer negotiation because there isn't a default MTA. I don't know if the ethics working group as part of the HCA is thinking about, I know they're thinking about standard consents and how to help people write consents to collect the data from donors, but I don't know if they're thinking about standard language to share biomaterials, but we could ask the ethics working group if they were.
I'm also on that working group as well, and I don't think we've touched on that yet. So
Fair enough.
That's the extent of my working groups, though. But while I still have the mic on, I've had another idea that I've been sort of pushing around for a while, and I don't know if there's general interest on it. It costs some money, so hence I can't really get anyone to bite on it. But I'm involved with the GTEx like deceased donor collections in the US, and I still have that network up and running. And we're actually sort of collecting some samples now for a couple of single cell and spatial studies, so fresh specimens going forward. And then I know the group in the UK who also have the deceased donor collections there. And I posed, I think, to CZI at one point, what about somebody who's doing some central funding of some highly collaborative efforts? Consider just funding the collection of a handful of donors with multiple core tissues from both sites and centralizing that as a resource where people can access those samples to try multiple different assays on them. And I think it requires doing it up fresh, but there's an appeal to it because we could standardize just getting some of the samples and the sharing of the samples as opposed to trying to piece together a lot of the back end. But I think both are very valuable, but I still think there's a lot of value in considering doing that up front as well.
Yeah, sorry, you should go ahead.
I was just going to say, I agree completely, Kristen, and I would say that one area where I've seen that work is instead of funding on the collection end, funding on the investigation end, to give somebody enough money to go to 2 sites and bring it into their technology and do that work and understand a little bit. Now it's just that investigator or that group that's doing that, but that's another way to sort of get money into the system to accomplish at least some of those goals.
This is Melissa. I'm just hearing a theme around funding, and seems like one of the outcomes of like the next steps might be how to think about funding models, short-term, medium-term, long-term, what would be needed for centralizing, having a stable, centralized biobank, as well as, you know, being able to fund specific projects and some of the other specific examples that you've brought up. It seems like a very common theme here. And one of the hurdles to actually being able to accomplish the actual sharing of samples.
So just one comment on kind of what you were just talking about, Kristen, is that's very similar to what CHTN does, that's funded by the NCI already, is to be a repository. It may not collect all types of samples and things like that, but they certainly have a lot of tissue, and they work with investigators across the United States, as far as I know, to produce both fresh, frozen, as well as fixed samples of both normal and diseased. So similar to, I think, what the interest is, but maybe could be expanded on with kind of guidance from others.
Oh yeah, schedule. Okay, so so I like what we're talking about in terms of a central repository and different levels. So one idea is to actually have physical samples coming in, and some, as Sarah was just saying, they're doing a little bit of that. Other HTN is doing some of that. It's hard to get funding, as Kristen was saying, but also this idea that you could do it on the sort of linking people together. If enough people contribute what they do have to a central place, the discoverability is another approach to at least putting people in touch with where the samples are. It sounds like that's perhaps more achievable in the short term, but it's also a little bit kludgy in the sense that the data is only as good as what people update you on. You don't know if you're a central sort of portal that's collecting information from people about what they have, eventually they will run out, and then that creates its own set of problems. So I think we have a little bit of understanding of what we think would be we would like to do in this area, that there's a need there's a want to have biospecimen sharing and discoverability, and that there needs to be a centralized metadata approach to that based on what the work's been done by a lot of you here on the call. In terms of touching on some other things, we talked a little bit about protocols, making them widely accessible, and pilot studies that would establish cross-consortium compatibility. This goes a little bit to what we've touched on around biosamples, is that it seems reasonable to me from a funding standpoint that if consortiums in general and their budgets make allowances, is one small way to do it, to actually fund people to do pilot studies. That would be a great way to go. It's hard to write a grant around something like that, but smaller projects that could be funded through other mechanisms might be a way to accomplish that. If anyone wants to add to that, please do.
Just from a being able to do things perspective, we are funded currently by CZI to do work around metadata standardization and around collection of even just sample records. So if other people are willing to engage with us to give us sample records, we can essentially already pay for some of the informatics components of these challenges. Obviously, doing so would come with we'd love to sort of use our proposed biosamples back end, but we'd be very happy to talk about details. And I think there are some very very low-hanging fruit that could be achieved in what we've discussed, at least from the sample metadata perspective. I mean, just as Kirsten said, there's even just getting everybody to ask the question. Which projects may have biosamples they could share, and even just capturing that on a project by project basis. If we can establish some kind of project registry, not even sample metadata, just these projects. Talk to each other, because you might have samples that you want, that each other wants, wouldn't be a bad thing. And we have started, although haven't managed to put make it a priority working with the two UK initiatives that I mentioned, the Addenbrooke's one, CBTM, and the Human Development Bioresource, to do this, to get that data into biosamples and make it more easily available, either for a perspective, if I wanted to do such a study, this group could get me the tissue, and from a discovery, they've already got it frozen, I can ask them for it.
Hey, you guys, Bruce Serna here. Nice to see all of you. Sorry, I've been working on something else, so but I've been listening attentively, and this is one of my favorite areas for all the reasons that Kristen and Todd and Laura have talked about. This COVID situation, it kind of brings up a painful example of why we need metadata across a whole lot of samples, and kind of given is semi-rare, moderately rare, anything from moderately rare to very rare situations where we've got to have deep metadata about samples that where we can look for sub-pattern correlations in subgroups of samples. That it just really vividly brings out why the sample metadata that Laura looks over at EBI is that rich of a model, is stuff that we need to have available for use. But when you go through the list of everything that's there, it's a painful exercise for users to really address all of those fields and categories. So it needs to be sort of an opt-in metadata model with some means, maybe under some scenarios where you get a linking ID to the sample such that you could follow up with it if you do find something interesting about it. I mean, that's a really neat option too to have. Don't know how feasible it really is, but I think all the reasons all the reasons for having multiple technologies and multiple accesses to a common sample set at anything, the specimen level or purified RNA level, DNA level, even accompanying it, is that's also a good way to group all the reasons and priorities for doing that. Let's say that there's a set of transcripts which are non-polyadenylated, and we need to get after that with a non-oligo DT based library generation method. That's the kind of thing where we need to have that metadata about the what the technology was, so it's not just like a batch and reproducibility of assay. It's a real technology shift that is going to let you see something different about the sample. Just some ideas there.
Todd, may I jump in?
Please.
Stavros here. I just saw in the chat that Marianne would like to, she's one of your scribes and would like to move to a different group afterwards. And also it is break time, so probably
Yes.
a good time to pause. It looks like you guys have been up to quite a bit of work. And so the task for part 2, if you wouldn't mind scrolling down to that blue box, is really just to, yep, there you go, is really just to kind of pick some of those key areas from up above and transfer them down here below, remembering to highlight the problem, kind of highlight any additional next steps that you think might be your recommendations for people to take down at the bottom. And this is what we're going to go through and harvest. Please feel free to duplicate this blue box as you see fit. If you want to have multiple of them, that is absolutely okay. Don't try to address everything in one.
Okay, so we covered, when I was looking back at at the discussion prompt, I feel like we actually touched on many of these things already. So we can focus on narrowing down our how might we questions. The only other thing I wanted to ask about, because it's something that I've thought about in the past, is what constitutes a biospecimen? Normally we're talking about tissue, but I wanted to see what the people think about things like cDNA libraries. For example, for single cell especially, a lot goes into making those libraries, and sometimes they can be sequenced deeper, or there's a reanalysis or another method. Is there any sense from people that that's a reasonable additional, more or less, biospecimen to be collecting and banking for other groups to work on?
I think that makes a lot of sense from my perspective, certainly. And I think it wouldn't just be narrowed to that kind of subsetting of a biospecimen. I think you could kind of RNA, DNA, and other, not just kind of whole specimens, could fall into that category.
Yeah, a quick note. I think it's a really good idea if people are storing RNA and DNA, sort of, because obviously they are much more stable. That's something that at least the HCA metadata model we would have to extend to accommodate, because we don't consider those things biomaterials. We essentially encompass the data collection represents that, we say. So, but it's not impossible. It would be a relatively straightforward exercise to add that as a type of biomaterial to how we collect data. But right now we don't do it, but that's, yeah, no reason not to. And it will be interesting to know how many people are doing that, because I don't know for single cell work how precious people are about having done the library generation themselves, because this is not like sort of back libraries where you pick a clone off a plate, so I don't know, do we have a feeling for sharing of libraries is actually something that is done so much when this is such a fast moving field from how you create them?
My guess is that for the reasons you said, Laura, that it's probably less so. I think people are very particular about their method being better. And RNA, obviously in GTEx, we have a residual RNA bank, which does get accessed a lot, but it's bulk tissue. So if you take it down to the single cell level, there's less RNA really there to share. DNA, I think if people want a DNA sample for an individual DNA sample, that may still be in demand. But I think it's getting maybe back to the cell or organoid resources where there might be more applicability for sharing and banking.
I think it's really valuable when you think about making scaffolds of a sample using some distinct technology, like long read or or non-Pallier dependent stuff. Kristen, where are you guys in GTEx, or where have you been on long read technologies and trying to get either Oxford or PacBio and really trying to get out isoform details and that could be used for a scaffold for secondary analysis of, let's say, single cell?
A little bit stymied by lack of funding, to be honest. So we have a very small data set of early PacBio, and we did want to sort of approach it to create some resource exactly as you describe. But the early data was not particularly good. We worked on it with Joshua Levin. And then with Dan MacArthur, they did a quite large project using Oxford Nanopore. And again, it's a very high error rate. So I think they'll release those data, but I'm not sure how good they are. And then recently, Joshua Levin's been working, trying to get some isoform structure, longer reads from single cell data using some GTEx samples, but we don't have a lot of it, to be honest.
I think both Oxford and PacBio have made some really significant improvements in their chemistries and a little bit of the informatics too, that are giving, I think, a much higher yield, lower price too, and less errors or methods to overcome errors.
So that would be on banked RNA then that you would use those.
Could
right?
be, or I mean, you could have it match up at different levels. So a bulk RNA prep from the same sample from which you made single cells, and then have aliquots at both levels, or you could from the single cells also have a sort of pseudo bulk RNA prep made before it's sorted out into single wells. So all sorts of different levels of crisscross and connectivity in the samples. And
so what I think I'm hearing is that you know, banking DNA is not uncommon, RNA less common but needed and accessed, and that the idea of doing single cell either RNA or actually the libraries themselves is not routine but could be useful for secondary analysis. Am I summarizing that correctly? No objections. And to accommodate that, it would probably be good. Again, I think you're right on the data collection or the sample collection labs. They have that. I know in my own group, we have our own single cell and single nuclei libraries that we save in the freezer. We want to go back to them. It's not uncommon to do a short run, you know, in fiscally constrained settings. We do a light run to see if it's going to be good enough to spend more money on doing a deeper sequencing. So we're accustomed to having those. And I feel like, you know, I've seen data where people in some cases have done much deeper sequencing to get more information and valuable biologically relevant information. Most of the time that's not done, but as sequencing comes down, it might be a viable way to go back in. So that's why I bring it up. And certainly technologies improve. In GoodMap, we're going back to do a realignment of the raw data from about 400 RNA-seq, bulk RNA-seq experiments. And what happens is, because technologies have improved so much that the quality control cutoffs are very different now. So some of those samples drop off or don't make the cut. And it would be nice to perhaps have done, in some instances for certain rare samples, to have gone back to the original RNA and perhaps reevaluate those. If it's okay with everybody, I think we can move into our blue phase and start to think about how we can narrow down some of these how might we questions. Is there anything that particularly sticks out to anyone that they want to make in terms of a priority list from this group of questions that have arisen for us?
So I've suggested two in the box below as kind of topics, and that maybe some of these questions fall under, and one around this idea of kind of developing some sort of shared biobank, and then the second one around what metadata might support that.
I was going to say what we might want to do is the metadata is vitally important, but I think the metadata schemas and ontologies group yesterday did come out with there needs to be some cross-consortium, and in fact there is already a cross-consortium metadata working group. It's sort of curation as much as metadata, but it's clearly an important part of it. And in the spirit of not reinventing things, we definitely need to give the use case to the metadata, the cross-consortium metadata working group, that we need shared metadata standards that enable sharing of these biomaterials, but we probably shouldn't reinvent a new solution to that problem. I think it's about feeding the use cases identified for sharing of biospecimens to that working group, rather than necessarily coming up with an independent entity.
That sounds like a great approach to that. So use cases would revolve around the idea of either having funding an actual physical biobank that also would involve using this metadata recommendation from the working group to facilitate sharing of these specimens, or as an alternative, perhaps creating a metadata only centralized registry of samples to make, so you know who has what available. And I actually, the idea that people have discussed with a project registry as well, I think is a very useful way to bring these things together. Any other comments?
Maybe one extra idea is just some concept of getting a in the registration process, the actual technology. We should invest more in a common technology, some sort of an API, let's say at EBI, that where you could get a reusable URI or something that then facilitated the kind of cross. If you got such a URI for a sample, you're essentially allowing a data model to be made from it that could be reused or searched from lots of different places. So it would be, yeah, for a central registry, and then you, however you're using the sample metadata in your system, it's derived from authoritative lookup of in terminology space.
Even like, I'm sorry, go ahead.
I would say, no, what Bruce is suggesting, and obviously if we were to use both or either the NCBI or the EBI biosamples databases, they do accession their samples, and they're stably accessioned. They're not versioned, but I do think they have update dates, and that's again something we could always address. And so yeah, having a stable URL to a sample. So then whatever services that wanted to build discovery interfaces on top of them or adopt that information. I mean, in other projects where we've supported tissue banking, they've even gone as far as the ID was assigned before the sample was deposited, and that ID was part of the barcode on the aliquot. Obviously, I think that's not necessarily something that would happen in the first instance here, but it's a useful sort of forward thinking.
It sounds like minimally it's minting a DOI, a DOI. Is that what it?
Kind of, yeah.
It's yeah. I mean, we have used accessions rather than DOIs, but again, you could, if you wanted to use the DOI model, that would also work. Again, it's just a permanent persistent identifier that is sustainably stored. Is definitely an important use case for shared biomaterials.
So it sounds like that would be registering a sample that you would like to share, or you want to share in a centralized portal. You would want that issued at that time.
Yeah, I mean, we actually already, well, we're not doing it with shareable IDs right now. We're just doing it internally for the HCA. But any data that's archived at EBI databases, whether the biomaterial is reusable or not, gets this stable identifier, because otherwise people can't persistently refer to it. And so that essentially is a solved problem. But it is important. It is an important feature of whichever solution we choose to use to share all this information, that solution needs to have some kind of persistent identifier associated with it.
So is one of the topics that we'd like to put forth the idea of potentially a collaborative grant application to build a physical biobank?
That was my question. Is it meant to be a one physical biobank, or is it meant to be more of this virtual biobank that really puts an infrastructure in place that allows biobanks that are in existence to kind of put tissues forward in a way that they could become shareable?
So my take on that is that they're two separate things. They're two blue boxes. One is basically, computational infrastructure metadata to find out what's there and to troll projects for what's available, where people might be able to do overlapping assays and add data. The other, which I proposed, is smaller. So it's not building a centralized biobank. And I think there'd be massive pushback to that because the sheer number of biobanks in the US, in the UK, elsewhere, in people's freezers is huge. And nobody's knows to this point how to unify them. But in the collection I suggested, what I was getting at was that we're at a point now where we have different groups trying to get very small precious samples to generate spatial image data, single cell data from. They have limited samples, they have different technologies and assays, and people are doing things differently. And to use the two resources we have, and there may be more, but the two I'm aware of are sort of the GTEx collection network and the deceased donor collection network in the UK, to just bank a set of samples that are for general use, where anyone who's trying or developing a new assay wants to benchmark that so that that data is required to go into one of these portals, can access those tissues. And then we have a way to sort of tie some of the benchmarking and tie some of the different analyses and experimental artifacts onto those and tie them together on the same samples. So if we did it from these deceased donors, we'd also have multiple tissues representing lung, kidney, something else collected across the same donor. And I think there's, and because they're deceased, we're not limited by the amount of tissue we can get. So we can get sufficient amounts and aliquots that it would serve multiple assays. Maybe there's no appetite for that, but I do think it would be a great resource and it needn't be terribly large.
I suppose that's maybe one of the things that it's worth investigating, what is already out there, because I know in the UK at least, the folks at Addenbrooke's are trying to essentially potentially turn this into a countrywide initiative, so other transplant centres can do equivalent research collection. I know the National Blood and Transplant Service already allows organs that have been rejected for transplant to go for research use. So a lot of the kidney data you get in the UK comes from rejected transplant kidneys. So it may be at least in the UK, in the UK and potentially in Europe, because these organisations already exist, it may be a hard sell to establish a new one, and therefore the virtual idea may be an easier way forward. I don't know with the NIH or sort of North American based funders whether there is a greater, whether it's so disparate that it's more, you know, there would be an easier sell to establish a new one.
Yeah, I think what Kristen's also getting at is a standardization in terms of collecting a sample. It's not any institution that has a rejected organ. It might be a limited number of institutions where you have a very controlled collection designed around this particular purpose and not the afterthought. Not saying that's not valuable, but eventually you'd want to have many different types of patients involved. But I think controlling the isolation is something that I've seen in other contexts is a very powerful way to limit the amount of variability in the samples so that you can discover the variability in the approach.
I guess it would be, given the variability into the approach doesn't seem to have, at least right now, hasn't dramatically impacted the community's ability to generate reasonable assertions on this data. It may be a hard sell to funding agencies. So it might be one of those things that you have to wait another couple of years to see how it plays out, with it not being done consistently, with more ad hoc resources, more sort of waste from biopsies and research, you know. And to be fair to CBTM, they are trying to do this in a consistent manner. They're not, you know, they're aware of experimental bias. They're not trying, this is not rejected transplant organs. This is collected in the window between the cavity being opened for the transplant organs to be taken and the cavity being closed so the deceased individual can be sort of buried, cremated, whatever.
Sure, yeah.
And so I suspect from at least a bottom up, the way the funders are operating around these things right now, a virtual biobank might be easier to get funding for than an initiative. That said, if you wanted to do an initiative like GTEx, where there was consistently collected data to do a benchmark and to say, okay, these 5 centers were doing this anyway, we got them all to collect the same data, we got them all to do it in the same way, these are the results. But that's different from establishing a biobank that is maybe an ongoing entity. But I think all of these things are possible, and it depends on where you are. My impression from UK and European funders is that because there are sufficient organizations that already exist, a federated solution and benchmarking experiments would be easier sells than a new biobank.
I think that's actually what I'm getting at, Laura. I'm not envisioning a new biobank beyond the fact that these need to be physically housed somewhere. But in the discussions we had at some of the CZI Seed Network meetings and others, to take the approach both in the UK and in GTEx, and to take a handful of core donors where you sample across the same tissues uniformly, maybe in 2 places, with a sufficient amount of material, and that it meets the underlying needs of some of the core tissue networks right now, so that the diverse array of assays have a single point to come to where those tissues were collected systematically, and they can all use their assay on the exact same tissue. Whereas going to an existing biobank, maybe 1 or 2 people can go and then the resource is gone. But to collect it so that it meets enough of the resources for spatial and cellular work. So not a big effort, but more a systematic resource that's biobanked for these kind of benchmarking efforts to tie in the different assays, so that there's some uniform set of samples that can span those efforts.
So I like the idea that these are 2 blue boxes. One, the idea of the virtual biobank that we've been discussing a bit. And I do think the idea of a systematic resource, as Kristen just stated it, for benchmarking on a small number of large amounts of tissue collected from a small number of cadaveric tissues would be a way to provide a broad resource that can cover many technologies from many groups at probably a reasonable cost. So not a lot of cadavers, but lots of tissue. So those would be 2 things. And I think that covers a lot of many of our how might we questions, actually, because one thing about building out a virtual biobank, it will put pressure to uncover some of the ideas on what is the metadata. What about the ethics working group and sharing biomaterials? What would these biospecimens include? Would, for example, the RNA, DNA feed back into that associated with, say we had a central resource for benchmarking? Would people generally agree or want to add to that idea that many of those are under that umbrella?
I think they are, yeah, I think I agree with you. They are under one or other of those umbrellas, but some of them remain sort of sticky issues. I think, the actual sharing of biomaterials, some of the things Laura brought up earlier, how do we know when something's been shared or when it's no longer a resource? The issue of sharing globally, the various different MTAs and consents underlying things. I think there's a lot of sticky, thorny issues in there, but I think they can be parceled into one or other of those 2 categories.
Let me ask about the idea of how to keep things updated. Laura, maybe you have experience with this, but or anyone on the call who may, that is it a good idea for I'd say it's a virtual biobank to serve as the go-between for the negotiation of all this, so that there's a tracking method? Or is it too much actual work that draws away from the virtual biobank's efforts to maintain all of that?
So I think it's fair to say the EBI's role in these sorts of things previously is just being about a sample record and potentially making sure the data model has a sort of stock element of it. So I mean, because we've done this for cell lines, particularly for pluripotent stem cells, and then essentially providing APIs that the people distributing the samples can update the stock, and there can be notifications when stock drops below a certain level, so you can, you know, create more of the cell line. I think if you wanted this virtual biobank to act as the go-between from an MTA and a legal standpoint, that's somewhere where you definitely would need funding, or you would need one of the biobanks, essentially representatives from each of the biobanks, to be able to step up and sit behind a sort of, I suppose, umbrella corporation type situation, so they could address the queries when they came in for samples that they held. Because I suspect all of these biobanks will have slightly different rules, because they will be in different physical locations and be subject to different funder requirements and national and international law. So the EBI has always held a sort of, I suppose, a data clearinghouse, metadata clearinghouse type of role in these situations. And we have, if we were to take on, or if someone was to take on the sort of contractual negotiation element of it, that is something that definitely would require paying for, because I think that's my external experience of people trying to negotiate MTAs is it is not a straightforward process.
Yeah, I feel it's a roadblock for sure. So my inclination would be to stay out of that hornet's nest in order to have something useful in place. So I think that would be the line to draw in terms of engagement.
Yeah, I mean, I think discovery, and you could incentivize the biobanks themselves keeping their records up to date by essentially incentivize people contacting you. If people don't know, if you're never going to update your stock levels, we just won't display them, and then people don't know whether you've got it or not. I mean, I know definitely when we've worked with the fetal biobank, they don't really keep a very small amount in stock, but obviously that gets used very quickly, and so they're never thinking about this in terms of being able to supply stored tissue to people. Instead, they've always thought about it more in someone's planning a project, they want to know if this resource can supply them with what they need. And again, I don't know for fresh tissue banks, that may again be mostly about this. This is not about actually supplying people with tissue. This is about helping build collaborations, because people know this tissue bank could work with them and may and will be able to collect tissue that they need in a way that they need it collected. So it's more sort of facilitating collaboration rather than actually supplying samples. And I don't know, I mean, Kirsten, from a GTEx perspective, you may be able to speak. I guess you guys had more tissue because you were doing whole body autopsies, how that worked out. But yeah, I do think for some of these more nascent efforts, ongoing storage is not necessarily their first priority.
Yes, I think for some of the efforts working with fresh samples, storage is not. For GTEx, we have loads and loads of tissue left over. Unfortunately, we stored a lot of it in a fixative that doesn't make it amenable to a lot of the technologies. But there is a subset that's amenable. We've been doing single nucleus work on it. It's frozen materials. It's not open access, but I think in general, people are now starting to realize that fully open access may not be achievable anyway. But yes, for one of the goals of GTEx was to create a biobank. So there is lots of stored material, which is why I know it's doable from these deceased donors to create sort of a benchmarking set.
Regarding the idea of the benchmarking set, do you have a sense from the GTEx effort on how many patients across what type of spectrum is a useful number of people to have included in terms of samples?
You know, if it's just you're not trying to recreate the study GTEx did, which was to look right across the spectrum of genetic variation. But let's say you just wanted to represent the core major tissues that were, you know, underlying some of the core efforts right now. I don't think you need that many. You need a reasonable amount of material so it can be shared across the different assays and preserved in a manner that's going to be most amenable to most assays. Obviously, anything requiring some fresh tissue isn't going to work. But anything else that can use stored frozen materials or OCT or fixed, you can cover those grounds. And that's what we're trying to do in some ongoing collections. I did float the idea to some CZI colleagues that both the UK and the US biobanks could deliberately target perhaps collecting some representative ethnic diversity within the UK and the US, which wouldn't be impossible, so that it wasn't all Caucasian white donors, and that might be a very useful set to have, maybe 10 or so of each, but it really depends on the question. I think having sufficient material for multiple assays across multiple organs within a donor is sort of the key goal, where those are collected systematically. And then the question would be, how many individuals do you want, just so you have some variability and can meet some goals without it being, without you trying to assess human to human variability, because that's not really the goal.
Mm-hmm. Who do you think are the people most interested in using such material for their benchmarking? Would it be a new project? Would it be a pilot study to go into preliminary data that you can do this on a massive scale? Is it consortium that want to set a standard?
I actually think that several of the, for instance, the CZI seed networks or some of the may use these materials, and it would tie them in. Let's say it would tie the heart people in to having done assays on the same individuals as the kidney people or something like that. So I think in the context of some of those seed networks, there could be widespread use. I think possibly for some of the HubMap assays, if they were available as a resource for the image-based assays that are being done in HubMap, along with some of the more single cell assays and chromatin assays that are being done in some of the seed network mapping efforts. But you know, I don't know. I think I think if the resource was there, several groups would use it. But it takes some effort to get the research off the ground. It doesn't take effort to say what it is we could do. The mechanisms are there, but it requires somebody, you know, the people doing hearts can say, I can get my hearts or this or that, but somebody saying maybe a unified effort would be worthwhile. And maybe it wouldn't, maybe no one's willing to fund it. When I propose it, there's always interest, but getting to the point of getting it funded is hard.
I mean, I don't know, I remember 2 years ago now, this sort of thing was mooted around figuring out how to record spatial information for the CCF, and that might be the crux of a clearly a clear problem that needs to be solved, that you could coalesce enough people around and convince a funder of, is that we need to include better location recording. We have a proposal on how to record locations we have these centres who are willing to do the collection and use the process, and then we want to see how it plays out sort of end to end. But you'd have to find the people who are willing to do it. And I think, right, again, Christina, you may be right. It's one of these things that everybody thinks is a good idea, but it's nobody's first priority. And that's true of both the scientists and the funders, because it's not going to make headlines and give them a front cover Nature paper in the same way as a lot of the other work that they're doing is. And so that might be the challenge, is getting enough people to do it. I don't know how much. Kirsten, you mentioned that the Standards and Technologies Working Group from HCA, right, I think they've done two, one which was around technology and one which was around sort of cryopreservation. Is that, this feels like the obvious next study for that group to do, but is it a study that group's talking about?
No, well, we have talked about it, but the problem is, sorry, I've got a phone going in the background, that the problem is, if we're trying to look across tissues, so that group has dealt with cryopreservation and things that are easy to get, like blood and materials you can purchase, because you can get a stock of them, which everyone wants, and share it among, and there's no shipping issues or different country issues or MTA issues. We'd love to move into tissues. So the first thing is, are there existing tissues we could do this with? And then we hit the MTA, shipping, different country roadblock. So if this resource already existed, that would solve that problem, but it doesn't exist. So, and that group is funded more towards standards and technologies at the assay level. So it would benefit greatly from having the resource, but it's not really funded to create the resource that it would benefit from, if you see what I mean. It's a bit of a catch-22.
So are we almost at the position that if we could create the virtual tissue bank, even if it was just a tagged set of entries in biosamples or something, then people might be able to do early sort of pilot studies where there wasn't any consistency of sampling, just to see what the diversity was without consistency, and then build an argument for the rest of it. Is there sort of, essentially, is the virtual tissue bank the low-hanging fruit in this problem space? Because all of the other problems have a sort of sociological hill to overcome. I mean, obviously metadata standards is still a sociological problem, but it doesn't involve engaging legal entities like MTA create sort of, and physical sample sharing does. Is that the first problem to solve, the sort of virtual sample discovery, rather than this other thing? And then we just have to keep advocating for what people have to keep advocating for what you're proposing, Kirsten, and it may be that we have to let the first round of data collection happen and be able to point out, actually, there's a bunch of unanswered, experimental batch effects. If then with a bunch of batch effects that nobody knows is biological or not, then you can make the argument for the we need to convince, get 2 or 3 whole autopsy samples in UK, in Europe, 2 or 3 European countries, in the US, and in Asia or wherever, then to do this but before it's been demonstrated that this is a large unknown introduction of error, it's difficult to make the convincing case.
Yes, it might be. I think if we can get samples that have data that goes into databases and get additional data generated on those and be able to link those, so that we know where they're there, and then when those data are out in the databases, we know that they're linked and we can compare some of those. I mean, if you think about existing efforts, we have these sort of ongoing HCA efforts with sort of separate sample collections, and it's not centrally funded, but there's several of those with the goal of sort of contributing to the HCA DCC. Then there is the centrally funded HubMap efforts. Obviously, we're here for HubMap, HCA, and all the other efforts collaboratively together. And HubMap within HubMap has this mandate to collect samples so that other image assays can happen on those. but it would be nice if those samples were to be able to be shared more broadly outside of HubMap, say with some of the other HCA efforts, because they overlap kidneys, lungs, other things like that, so that we could generate a broader set of data. So maybe within these existing efforts, and likewise, there may be individuals producing data that they plan to submit to the HCA DCC that have residual samples, that it would be really good to provide to some of the HubMap investigators to see if we could get image assays on them. So yeah, I mean, that may be the starting point, and that may be the collaborative thing that we push in this meeting here, where we have these investigators here.
That sounds like
go ahead.
I was just going to say, that sounds like a great plan for a use case or pilot, to take what you were just saying, Kristen, about starting with what we have and moving that forward to see what you can be shared. I'm just thinking towards like what we can, you know, when we Sarah's already been filling out our blue boxes, so just thinking about, you know, next steps. I was just thinking about what we might want to bring forward from this discussion into our recommendations for what we want to do. Go ahead.
I was just going to say another area of incentivizing, if you will, around a benchmarking study is if samples are available both within a consortium and beyond a consortium. If you're going to do a pilot experiment and for applying for a partnership grant with that consortium, or evaluate your technology for inclusion in an existing consortium, using a benchmarking sample is a great way to tie in very clearly what your capabilities are and make it relevant, maximally relevant to what's going on. So virtual followed by, that would promote the idea of the benchmarking studies or samples.
Yeah, I just put some suggestions up here, please. I welcome people to edit kind of the content of what's in each of them, of things I might have missed or extra text that maybe is extraneous.
Well, beyond these two blue boxes, is there anything additional that people feel is an important thing to bring forth from our discussion?
So I just, oh my, no, I am muted. I was just interested in looking at Chi-Storm, and it seems like in the soapbox sign up, the folks who are in the 15A and 15B have put up a proposal to on the soapbox to talk about combined efforts for biospecimen and protocol sharing and overcoming the barriers. So I do wonder if they've ended up having more or less exactly the same conversation as we've had. And it may be worth touching base with the moderators of those other two sessions to try and end up making sure we're proposing the same thing and not two different things.
Yeah.
Hi, folks. That was me. This is Gloria Priover. Hi, I was the moderator for those two sessions. I let our groups go a little bit early for lunch. But yes, we did. I'm just looking over your documents too. There are some similarities. We focused on, in terms of sharing samples and the idea of biobanking, the first piece that we got kind of stuck on was the legal and institutional barriers for sharing. And that got us to consents and got us to the idea of perhaps getting a group together for a common to write a common consent or common consents across consortia that we might then, the way I put it was to have the NIH blessing on those consents, that then our IRBs and institutions might be more compelled to, be able to sign off on the sharing. So that was one, and the other was the protocols. Again, we talked a fair amount about having a common place for protocols to be placed and accessible, and not only that, but to have a way to give feedback in real or near real time, right? What's working, what's not working, comparing the protocols, what analyses they work for or don't work for, making suggestions to the developers that might move those protocols along. We talked a bit about protocols.io, and then we came down to, somebody just needs to make a decision about it. So optimally across consortia, that we might make decision about what we will use cross consortia as a place to deposit protocols.
So just to touch on your first point, we discussed briefly a little ourselves. The HCA has an ethics working group, and they've been undertaking a huge effort to produce draft consent templates for different circumstances that can be generalizable globally. Mm-hmm. And that's being done under the auspices of the HCA. I think my understanding of HubMap getting going is that it's encouraged people to make sure they have genome data sharing and open access. I think the open access has pushed back a little bit as people were unable to do it. But again, there are some efforts going on there, but that seems to me to be, since I'm vaguely involved in both, seems to be roughly siloed in the HCA. So to broaden those efforts and to include other groups and to see what that synergy is, I think would really get to your first point. So there are some efforts underway, but let's expand on those and collaborate on those so that we can address that.
That's exactly what we want to bring out and get people talking from the various consortia together.
Gloria, we also, things that are a little different from what you have proposed, which now that I read them seem more distinct than I at first thought. We did talk about a virtual cross-consortium biobank, a way of registering samples as a discovery mechanism, mm-hmm. And a standardized metadata. We did, though we touched on a little bit of MTAs and things, we kind of realized that was going to be very difficult to navigate, and probably would avoid some of that unless, as you're pointing out, some leadership from NIH would help. I know that in some of the projects we've been involved in, that just the IRBs for collection for a large project can take forever, and MTAs are also a sticking point. So it would be good to standardize. There is an idea of the centralized IRB, for example, that's coming forth, that's getting better and better. So, but a virtual cross-consortium biobank, and we also discussed the idea of a more or less a benchmarking reference set of samples, which would be a small number of, say, deceased donor tissue, but a lot of tissue mm-hmm. from many organs within a sample, so that it could be used as a source for people to do benchmarking of their new technologies or existing technologies across consortium. Anybody would have access to it. Mm-hmm.
Those sound like really good points to me. Are you going to bring them to the table?
Yes, we're gonna, those are our two blue boxes.
Great.
I wonder if there is going to be a presentation at the sort of soapbox session at the end of today about this breakout. It seems like essentially we've got while we've essentially come to the same conclusions from different places, Gloria, maybe you and Todd talking about them together and saying it's from the whole breakout rather than just the one, because I think I think unsurprisingly we've had more or less the same conversation in both rooms. But I think the fact that we had the two rooms and have come to these conclusions independently is great evidence that they really are the pressing problems.
Yeah. So I'm hearing kind of 4 different ideas to carry forward. One is tackling the ethics and legal ideas or problems. The other is the protocols. Another is the development of a virtual biobank. And the fourth is the development, identification of sources or a source of benchmark tissues.
Mm-hmm.
What did you feel was your next step for the things that you talked about here? Is it identifying people, getting people to step forward and say, yes, we're going to carry on these conversations, or?
I think that's definitely the case for the virtual biobank. Would the rest of my group agree?
Yeah, that seems like we need to establish, I think, an important point that we made, that there are clear metadata standard definitions needed, and we shouldn't circumvent the existing Cross-Consortium initiative around that. Mm-hmm. But if we feed use cases to them and ask them to give us the standard, and then we essentially, wherever is the central repository for those samples, in sort of endeavor to make sure it can adopt it, and then we're in a good place to potentially even set up some kind of pilot without having to apply for funding, and then we can demonstrate it works and then convince the funders that if they gave us resource, we can make it even better.
Mm-hmm.
And then for the second part, Gloria, the reference tissue, that's more of developing what it would be and identify the benefits of it. Mm-hmm. And that would require funding. So that would be somewhere where we want some people to come together collaboratively to define what is needed and then apply for funding from